You're good.
Okay, it looks like we're recording. So I'm going to be going over 1 and 2. So in letter A, which of these terms describes or is an example of a regulatory protein? We have activator, repressor, and cap. Which of the terms describes or is an example of a small effector molecule, allolactose, inducer, and camp, or cyclic AMP? Which of the terms describes is an example of a sequence found within the DNA, which would be +1, -10, and the operator. D, which of the above is not involved in the lac operon, which would be inhibitor? And then finally, which of the above is an example of a structural gene, which would be lac Z and A, and also Y is there as well, but it's not in the list. Fill in the blanks. The lac operon is regulated by 2 inducers, allolactose and cap. The binding site for sigma factor and RNA polymerase is called LAC-P, while the cap site is the binding site for the activator that regulates the lac operon. The enzyme adenyl cyclase converts ATP into CAMP. The -35, -10, and +1 sequences found within the LAC-P portion of the lac operon. The LAC-I gene encodes for the lac repressor, while the LAC-Y gene encodes for lactose permease. Attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And an E. coli strain that contains 2 copies of lac operon is called a merozygote strain. So for 3, we're going to be having Joshua go and describing this.
Cool. So for this one, it didn't explicitly say whether we have lactose, so my assumption is that we have no allolactose, so we use IPTG. All right, so I did. And so for this question, each of these is asking whether we have blue E. coli cells, and these would be colored by X-gal molecule. So in the first A, we would have colorless cells, so no blue cells because glucose and IPTG are absent. Therefore, IPTG doesn't pull off the repressor, and transcription of the lac operon can't happen, even though X-gal is present. For B, glucose is absent, but IPTG and X-gal are present. IPTG helps release the repressor, so lac operon can transcribe X-gal, then makes the cell blue. And C, no cells are blue because X-gal is absent. And D, glucose is present, so no transcription of the lac operon occurs. And transcription only occurs, if you remember, when glucose is absent and lactose is present. And then for E, it's the same as C. No blue cells are present because X-gal is absent. And now for question 4, we have Nathan.
All right. So, that's a loud bang. Someone just ran into a wall. A is inducible because all the genes are functioning and present. B is non-inducible because the repressor will be stuck on the operon and immovable from that position. C is constitutive because the repressor cannot bind to the operon, so it's just always going to be transcribed. C is constitutive because that's what I just said, C. D is non-inducible because the broken repressor, though only on the plasmid, will eventually take up all the binding sites. And E is constitutive because the chromosome will be non-inducible on the, so the gene on the lac operon on the chromosome will be non-inducible, but the plasma is constitutive, so the lac operon will always be being produced by the plasmid, even though the chromosome can't. So the constitutive takes precedence there. And then for number 5, in situation A, you have the defective INP, and so in the presence of lactose, you will have 1, and in the, so it's constitutive, so you have 1 and 1. For B, similar situation, just with the operon, so you have a 1 and 1 there as well. For the situation C, the chromosome is inducible and the plasmid is constitutive, so the plasmid will always be on whether lactose is present or not, but the chromosome will only be on if lactose is present, so you have 2, 1 there. And then for D, you have, let's see, so you have essentially 2 broken parts. The chromosome has a defective repressor, but the plasmid has a defective promoter. And so those 2 broken parts together, though, makes it so that it is overall inducible. So you have 1 being created in the presence of lactose and 0 without lactose. And then E is non-inducible overall, and so you have 0 and 0. And that is week 12.